Now that you've become more familiar with genetic analysis of haploids, it's time to look at one example of an epistatic interaction that we'll talk about in haploids, this idea of suppressors. Suppressors are mutations at a second locus which act to mask a phenotype at another locus or suppress that phenotype. In diploids, we've talked about dominant epistasis and recessive epistasis, but in haploids, we don't talk about dominance or recessiveness because we've only ever got one allele of any given locus. So it's very easy to screen for suppressors. Typically what one would do is take some mutant, say an auxotroph of some kind, and expose it to a mutagen. A mutagen just increases the baseline mutation frequency so that we have a better chance of finding mutations in the next generation, in the progeny. And then typically we would plate on minimal media. Now, an auxotroph is not going to be able to grow on minimal media. However, if there's some sort of mutation event which changes the phenotype to prototrophy, we will see colonies that grow. So only prototrophs can grow on minimal media. And now what we've got are things that were once auxotrophic, they've been exposed to a mutagen, and now they've become a prototroph for some reason. So there's two general possibilities, two options that we'll discuss that could underlie this event, which is called a phenotypic reversion. The first option is something called a back mutation. A back mutation is simply fixing the problem at the original locus. So, for example, if this was, say, a methionine auxotroph of some kind, we'll call it MET1 minus. A back mutation is simply a conversion from MET1 minus to now something that is fixing that original locus and reverting it back to a wild-type phenotype. So now we've got a functional copy of the MET1 locus. So that's the one option, fixing the original problem at the original locus. The other possible way to get this phenotypic reversion is this idea, what we've just been talking about, a suppressor, having some second mutation which now masks the effect of the first. If that's the case, we would have a conversion from MET1 minus, and the mutation event would not change that original locus, it would still be MET1 minus, but now there would be a suppressor involved, which I'll abbreviate just as SU. The wild-type version of the suppressor obviously has no masking ability because that's how we found the original MET1 minus mutant in the first place. Whatever this is, it's not able to mask the deficiency at the MET1 locus when it's in its wild-type form. But the mutagen has converted this allele from the wild-type allele to some sort of mutant allele, and this mutant allele is now capable of exerting epistasis over the MET1 minus mutant. The big difference as we go on and look at questions that involve these different possibilities, the big difference here is that in the first case, in a back mutation, we've actually fixed the original problem. In a suppressor situation, we have a second locus, which is now mutant, which is exerting epistasis over the first. So the MET1 minus mutant is still present. And deciding the difference, or deciding between one type of event, a back mutation versus a suppressor, hinges on the ability to detect that this MET1 minus mutant is still present even after this phenotypic reversion event has taken place. So in the next segment, we'll talk about how we can do crosses to tell the difference between this type of event with a suppressor arising and compare and contrast it with what happens with a back mutation and how we can distinguish those. Okay, so here are the different options. You have a phenotypic revertant on your minimal media plate after exposure to a mutagen. And the different options are we had a back mutation that fixed the original auxotrophic mutation, or we've had a mutation at a second locus where this allele is now masking what's happening over here, so that both of these are now prototrophic. So they're prototrophs, and previously they were auxotrophs. So that's that phenotypic reversion. We've gone from an auxotrophic state to a prototrophic state. These are the two genetic options to explain that phenotype reversion. The key to distinguishing between these in experimental crosses is to cross each of them to wild type. We'll do the first one here, the back mutation. This is the simplest case. Because this is now a wild type allele of this locus, if we cross this with wild type, and again, wild type is just MET1. Wild type is just MET1 plus. This is just a cross of MET1 plus times MET1 plus, and the key here is in a back mutation case, you will see all prototrophs in the progeny of that cross. With a suppressor, because this MET1 minus locus is, or the MET1 minus allele is still present at the MET1 locus, we'll see that we don't get all prototrophs. There's opportunity for recombination. So in this case, what we have is we have the revertant, MET1 minus SU. This is the suppressing allele, crossed with wild type. And wild type in this case is simply MET1 plus, and the wild type of whatever the suppressor is. So it's plus plus with the crossed with the suppressor allele and MET1 minus. The transient diploid in this case will be a dihybrid, and then there will be the 4 possible genotypes of the progeny. So let's just sketch those out. There's the 2, we'll do the 2 non-recombinant ones first, MET1 minus SU, MET1 plus, SU plus, and then the 2 recombinants. Okay, now it's time to think about what phenotypes these different genotypes will have. Whereas here, in this first cross with the back mutation, everything here is simply MET1 plus, and thus prototrophic. Here you can see that there's going to be some that are auxotrophic. Okay, first off, there's the wild type one, so that will be prototrophic. MET1 minus with the suppressing allele is also prototrophic. That's the genotype of the original revertant. So this is the allele that's capable of masking this defect, so this will be prototrophic as well. Let's see, let's do this one here. This one, it has the mutation that gives methionine auxotrophy at MET1, and it has a wild type allele of the suppressor, which means this is not an allele that's capable of masking what's going on here. So this genotype will always be auxotrophic. There's no other possibility. It has the original mutant and nothing that's capable of suppressing it. This genotype is exactly the same as the original auxotroph, where we had a MET1 minus and a wild type allele of a suppressor. So this is auxotrophic before it went and was phenotypically reverted by this mutation which masks what's going on here. Now this last category is something that we just can't tell. It has the wild type allele of the MET1 locus, but now that wild type allele at the MET1 locus is in the presence of the suppressing allele. So this is the allele that's capable of suppressing the MET1 minus mutant, but we haven't seen this genotype before. And it's possible that this mutant allele, which acts as a suppressor of the loss of function mutation over here, it's possible that this mutant allele actually somehow interferes with the wild type function. Or it's possible that this would not interfere with what's going on over here. So we actually can't tell if this is going to be prototrophic or auxotrophic, and you have to examine the data in order to determine what phenotype it has. Now, in the next segment, what we'll do is we'll discuss how it's possible that this suppressor allele could somehow potentially mess up the function of what's going on at this locus, and that's a special type of suppressor, and we'll discuss that next. Okay, I've drawn these genotypes out again, and again we're asking this question. What phenotype will this recombinant genotype have? Remember, this is one of the non-recombinants here. This is another one of the non-recombinants. In the case of any suppressor, there will always be a recombinant that puts the original mutant allele in the presence of the wild type of what the suppressor, the wild type allele of the suppressor locus. So because of that recombinant, there will always be auxotrophs if a suppressor is involved once we do a cross to wild type. This other recombinant also puts the suppressing allele in the presence of the wild type allele of the original locus that was being suppressed. So the question is, what phenotype does this genotype have, and what can we infer based on that phenotype. The most usual case, or the most straightforward case, is when this has a prototrophic phenotype. In this situation, if this has a prototrophic phenotype, it simply means that this mutant allele is not interfering at all with the wild-type function of this locus. And that's the usual case. Usually, most suppressors don't have the ability to somehow interfere with the function of the wild-type allele of what they suppress as a mutant. And we'll talk about how that can now work, why biochemically most of the time suppressors don't interfere with the wild-type function.